So we continue our recording or our lecture on the use of DNA replication enzymes. And we're talking about industry applications, and this is part 2. And so let's talk about topoisomerase. As just a reminder, topoisomerase is the enzyme that goes in front of the replication fork and makes sure that it is relaxed in such a way that the replication fork can continue to proceed through as the helicase begins to open up progressive regions of the replication fork. And so this is the mechanism of topoisomerase, and you have a supercoiled DNA right here, and topoisomerase is going to bind to in front of the replication fork, and it's going to cleave, and it's going to relax and re-anneal. And so you go from 4 or 3 supercoils to 2 supercoils. In other words, it's relaxing it so that the replication fork can proceed through. So what is the mechanism? Well, the mechanism is that it actually requires, it's a bit complex, and we won't focus too much on that, but it requires 4 different amino acids in the binding site for the topoisomerase and the substrate. And it requires tyrosine, 2-arginine, and a histidine. But the bottom line is that the tyrosine OH then binds the phosphate group, okay, to kind of nick the DNA strand by binding to the phosphate group, and there's that free OH group right there, and it begins to unwind or relax the DNA by threading the free fragment through, and then it begins to ligate it again. And once it ligates, the number of supercoils is going to be different. It's going to be less. And what I want to point out, because this is very, very important, is that you remember in RNase, in the presence of alkaline, you remember that the 2' OH, which is not present in the DNA, so it cannot serve that purpose, but the 2' OH in the ribose of RNA serve exactly the same purpose that the OH on tyrosine is serving. And I want you to look at it this way, that it actually binds to the phosphate group. So this is the 2' OH, binds the phosphate group right here, and I want you, it's kind of backward, but the phosphate group is still attached to the 3' OH on that ribose right here. So I want you to keep that in mind. The OH, just replace this OH with the one on tyrosine, and so that OH binds the phosphate group, and now the 5' end of that DNA fragment right here is actually a free OH and not a phosphate. This is very important because when we talk about the application, this is very critical, okay? So the OH binds to the phosphate, and the phosphate is attached to the 3' OH on the nucleotide preceding the nucleotide that is after that. And the nucleotide after that has an OH on the 5' end, whereas the nucleotide before that has a phosphate on the 3' end. So that's very, very important, because when we go to the next one, and that is, well, let me talk about Taq polymerase. So topoisomerase then relaxes the supercode in the DNA so that the replication fork can proceed. Taq polymerase is a polymerase that is used for PCR, and you can see that it is used for amplification of template in the presence of 2 primers to amplify both strands, and the deoxynucleotide triphosphates. And the Taq polymerase, it can amplify any DNA template with a specific region or length that you set by generating the 2 primers. And it requires the use of three temperatures: a denaturing temperature about 94 to 96 degrees centigrade, an annealing temperature which is roughly around 68 degrees, and an elongation temperature which occurs at 72 degrees centigrade. Okay, so the difference between these and the helicase dependent amplification, the HDA, is that HDA, all of these take place at 37 degrees, whereas this one takes place at three different temperatures: at a denaturation temperature, annealing temperature, and an elongation temperature. So here are some specifics about the Taq polymerase. It's a single unit protein that is isolated from an organism called Thermus aquaticus from the hot springs. These are microorganisms or bacteria that actually live in the hot springs, and it makes sense because they had to have their DNA copied, and if there was a strange enzyme that will withstand the temperature of the hot springs, it would be in this microorganism, and that's where they discovered the Taq polymerase. Taq polymerase has a 5' 3' exonuclease activity, just like DNA polymerase 1, and then it has 5' 3' polymerase activity, just like DNA polymerase 1, 2, and 3, and then but it doesn't have any 3', 5' exonuclease activity or the proofreading activities that DNA polymerase 1, 2, and 3 have. Okay? The fidelity, though, is one mutation out of 9,000 bases. So this is incredible. The fidelity is incredible. But one of the unusual activity that is associated with Taq polymerase is that it adds an A to the 3' end, and that activity is actually exploited in cloning, and especially in the cloning, topoisomerase cloning, TA cloning that we're using in the lab. Topocloning. Well, topocloning consists of an open vector that is covalently linked to topoisomerase enzyme at the 3' end with a phosphate group. Again, you remember I said this is strange, but it's linked to the 3' end through the hydroxyl group on tyrosine, but it links to the phosphate that is located at the 3' end of the vector, one of the strands of the vector. The vector has a 3' T overhang, because you remember Taq polymerase generates a fragment that has a 3' A overhang on the PCR fragment, and the two ends are complementary at the 3' end. So they fit together, the overhang T, the overhang A, they exist in hydrogen bond. The 5' OH of PCR fragment then, so remember, PCR fragments are not phosphorylated, okay? So the 5' free OH group then is going to be used as a substrate for the topoisomerase, which can provide the phosphate that it binds to through the hydroxyl group on the tyrosine, okay, side chain. So the 5' OH of PCR fragment provides the other substrate for the topo enzyme to swap the phosphate back from the tyrosine OH to the 5' end of the PCR fragment. So by design then, the OH which grabs the phosphate group that is attached to the 3' end of the vector is going to grab the 5' end of the insert with the free hydroxyl group, and it's going to use that as a substrate to form a phosphodiester bond. And so this is what this shows. You remember this is a 3' end, right, with the overhang T, and this is a 3' end for the insert, 3' end for the vector. You can assume that the vector goes here, and then this is the 3' end of the other end of the vector, right? And then this is the 3' end of the insert and 3' end of the insert. You can see that T and A are complementary to each other, okay? So the tyrosine then the OH is bound to the phosphate, which is bound to the 3' end of this vector fragment right here. But that's okay, because the topoisomerase is going to use that free OH right here from the 5' end of that PCR fragment as a substrate to interact with the phosphate group that is now being released from the tyrosine OH, thus forming a ligated product into it. Okay, so you have the vector plus the amplified insert that is now ligated into the topo vector, and the end result is that when this reaction takes place, then the topoisomerase then falls off the vector, and so now you can take that plasmid, transform it into E. coli, and have the E. coli amplified. And this is just another way of looking at it. You have the vector right here, the ends of it, and topoisomerase binds to the 3' end phosphate of the vector right here, and it uses the 5' OH of the insert, 5' OH of the insert as substrate to form the phosphodiester bond, which is right here. Okay, so now we have a plasmid, and the topoisomerase enzymes are now released from that, and so you can do the transformation and amplification. Okay, now let's talk about telomerase. Telomerase is that enzyme, and we never really discuss it, but you remember that when I want you to imagine it, and if you have a hard time imagining this, I want you to actually draw it out on a piece of paper. So here's what I would like for you to do. I would like for you to draw 2 strands, DNA strands, running horizontally. Okay? Now, you remember that if the DNA strand that ends in 3' end, when you separate those, you're going to lay down an RNA primer, right, at the end that is complementary to that 3' end. RNA primer is laid down, and then DNA polymerase 3 comes in and begins to synthesize the complementary strand in a continuous fashion, right? Now, that RNA primer has to be removed. It is removed by the 5', 3' exonuclease activity of DNA polymerase 1, okay? But here's a problem. Here's a problem. When that is removed, there's not a DNA polymerase in the world that can fill that gap in. Here's the reason why. DNA polymerases that we know of cannot synthesize DNA de novo. They have to have a existing primer, and they have to have that 3' OH to begin to add nucleotide onto it. And so, for that reason, for every cell division, there is that gap that is at the 3' end of that DNA, okay? And so, if you're not careful, the end of the chromatids will begin to be shortened with each cell division, okay? Now, it turns out that in embryonic and stem cells, there is an enzyme called topoisomerase. And what topoisomerase is a reverse transcriptase. Now, look at the gap right here. At the end of that DNA fragment, or the telomeres, right? There are repeating sequences of TTA, GGG, TTA, GGG. These are just repeats that goes on and on until the end. And the reason why this is important is that because they don't code for any gene, any loss of this repeated sequence is not going to affect transcription of genes. Genes are here upstream from here, right? But you have this repeated sequence at the telomeres, and as long as you don't touch the genes that are interior to that, they will not affect transcription or copying of these genes. But you don't want to shorten these repeats too much. So the telomerase enzyme, which is present in embryonic tissue and rapidly growing tissue and in stem cells it's found, but it's not found in somatic cells. Okay, what it does is it carries its own RNA primer, and that's very unusual. It carries its own RNA primer that is complementary to the end of the repeated region right here. So it will do it will attach the RNA primer, and then it will begin to add nucleotide that is complementary to that. You remember, reverse transcriptase is an enzyme that synthesizes DNA from RNA. Well, here's the RNA primer that it is used right here. It kind of attaches to the end of the of this DNA strand or the telomere region, and it begins to add complementary DNA nucleotides right here to extend that DNA. So you extend that TDA, GGG, again TTA, GGG. So as you extend it more and more, now the RNase the primase can come in, lay down the RNA primer, and then DNA polymerase 3 will come and extend that. DNA polymerase 1 comes in. And so you see the point, right? When you have lengthening of this, then the machinery can work. The primase can lay down the primer, DNA polymerase 3 can come in, DNA polymerase 1. And so it kind of prevents the shortening of that region right here. And that's the action of the topo or the telomerase. Okay? And this is the function of the reverse transcriptase. You have an RNA right here, which is what you did in the first strand synthesis. Okay? Before you did the PCR, so you had the RNA, you isolated total RNA, you had gene-specific primers that were added right here, and you begin to reverse transcribe, and that is you begin to synthesize DNA from that RNA. So you have an RNA template for the reverse transcriptase, and then you have a DNA primer, and then you can extend that DNA primer by adding in deoxyribonucleotide triphosphate, and you can add each one of them to this right here. So this is the first strand synthesis. You have the RNA that is complementary to the DNA. This is the first strand synthesis, and from here on you can then amplify by PCR, and so that's when the PCR comes in after the first strand synthesis. Now, any DNA or any gene that you generated through reverse transcriptase or from RNA, you actually designate it by putting a C and DNA after that to mean that you actually clone it from the RNA instead of from the genomic DNA. Okay? Now, this is the action of the reverse transcriptase. Since messenger RNA, we could have done this. We could have isolated messenger RNA. We just did total RNA, which contains all the types of RNA, but we can specifically enrich the messenger RNA and use a poly DT primer, and you can extend every single RNA that is in there for first strand synthesis, and then you can come in and amplify specific genes by using gene-specific primers. But what we did was we simply isolated total RNA, and then we amplified the RNA by using gene-specific primers and so forth. Okay? And the final enzyme that I want to talk about is T4 DNA ligase. And T4 DNA ligase is used in cloning in the industry. And what it is that if you cut two fragments of DNA with the same enzyme, let's say you cut with UcoR1 or BAMH1 or SMA1 or PST1 or PSD1, the sticky ends, all right? The two ends will be complementary to each other. Now, they can form hydrogen bonds, but there's no ligation, right, involved. You need to paste in, or you need to attach the OH, 3' OH, and 5' phosphate together to make it a complete chain. And so this is a 3' OH. It's going to bind to the 5' phosphate by the action of the T4 DNA ligase. And same thing here. 3' OH, 5' phosphate will combine to form a complete double-stranded DNA. And that's what happens in recombinant DNA. Dr. Garner is going to talk to you a little bit more about this.